Hello everybody, I welcome you all to NCAS Geology class. Today I will discuss on the topic that is stratigraphic correlation. Earlier I have discussed on stratigraphic principles, stratigraphic elements, geological time scale, and now I will discuss on stratigraphic correlation. So what is stratigraphic correlation? Correlation is a process of establishing the equivalence of stratigraphic units or rock units which are found to be distributed widely away from each other, or they are placed in distant areas and their equivalence is being established through stratigraphic correlation. So how this equivalence can be established? That can be established on the basis of the study of lithology, on the basis of fossil content, on the basis of absolute age. Accordingly, the correlation of stratigraphic units are classified. So let us discuss on the stratigraphic correlation. So, correlation in stratigraphy is a process of establishing the equivalence or relationship between rock units, that is bed, formations, groups found in different geographical areas to determine whether they are formed during the same geological time or they represent same depositional event or environment or same stratigraphic position. Let us say A and B are the two rock groups which are widely separated from each other or are occurring in adjacent regions. In that case, following cases arise between these two rock groups. That is, A may be younger to B, A may be older to B, or A may be equivalent or same in age to that of B. So when this third relationship is being established, in that case it is said that the A has been correlated with B. So here the correlation is established. Thus the aim of stratigraphic correlation is to establish the equivalence among the rock units from different parts of the earth's surface and build continuous history of the earth. So what is the main aim of stratigraphic correlation? Main aim is to establish the history of evolution of earth's rocky layer in different sectors of the earth. So correlation is really helpful to know this history, for which importance is being given in stratigraphy to know about the stratigraphic correlation. So let us know what are the different types of correlation. These are lithostratigraphic correlation, purely on the basis of study of rock types and their stratigraphic position. Biostratigraphic correlation, which is purely based on the study of fossils on the boisons. Then chronostratigraphic correlation, it is based purely on the study of the absolute age of the rock units, more or less absolute age. And now we will find out the difference between homotaxis and lithostratigraphic correlation, because they are more or less found to be synonymous, but it is not exactly synonymous. Homotaxis means it is a combination of two words, homo and taxis. Homo means same, taxis means order. So homotaxis means the arrangement of the rock units in a definite order or the same order of lithologic units or their fossil content, their vertical succession and they are not necessarily same in age. So that is, we may find, for example, sandstone, shale, limestone in one place, and in another place also we will find sandstone, shale, limestone, but their lithological characters are not exactly identical. Rock types may be same, but they are, if you analyze their mineralogical characters, chemical characters, on those bases they will differ. But as the rock types are same, they are placed in same order, but they are not of same age. In that case, they are said to be homotaxial. For example, Kodappa and Vindhyan group of rocks both contain sandstone, shale and limestone, but their age are totally different. When Kodappas are older, the Vindhyans are younger in age. And again, if we take into account the lithological characters based upon chemistry and mineralogy, they will not be exactly identical. That's why they are said to be homotaxial. Similarly, Gondwana basins of India, the Damodar basin, Godavari basin, Mahanadi basin, they contain the litho units which may be identical, which may be placed in same order, or lithological units may be placed in same order, but they are not of same age, and their exact chemical characters, mineralogical characters may not match. Similarly, in case of Kaladgi basin and Bhima basin. So, in case of lithostratigraphic correlation, the same formation or member is found to be traced from one region to another. The units may extend laterally and are physically the same body of rock. This fact we have to give importance. That is same body of rock regardless of age elsewhere. Age is not taken into account in lithostratigraphic correlation, but rock types must have to be same, they must be placed in same order, and they must be identical with respect to lithological characters. In that case, it will be correlated on the basis of lithostratigraphy. But when you talk about homotaxis, there will be simply same order of arrangement of lithological units or fossils. And the age is immaterial. Age is not taken into account. Same thing is there in case of lithostratigraphic correlation, age is not considered, but the litho units must have to be identical in with respect to all characters. For example, Barakar formation. Coal measures of sandstone is traced across Damodar, Son Damodar, Mahanadi and Godavari basin. So it is the same lithostratigraphic unit which is correlated based upon lithology and mapping in these horizons. Now let us discuss in detail what is lithostratigraphic correlation. It is achieved by comparing and matching the rock formations in different geographical areas based on their lithological or rock characteristics and stratigraphic position, and not by age or by fossils directly. Let us say locality A and locality B, we find the same lithology, red sandstone, shale, coal seam. Here is red sandstone, shale and coal seam, but only difference is that in locality A the beds are thinner, whereas in locality B the beds are thicker. So the above two rock successions present same lithological characters, or they present identity with respect to their lithological characters, or distinct lithological characters which match, and they have the same marker horizon, that is coal seam, at their bases. So coal serves as the marker horizon. Coal is at their bases, which identify the stratigraphic position. Thus, these two rock formations are correlated lithostratigraphically. So, in order to correlate the rock units on the basis of lithostratigraphy, detailed study of lithology is required to establish the equivalence and then to correlate them on the basis of lithostratigraphic characters. So, in order to establish lithostratigraphy, the lithology, the order of superposition, thickness characteristics, lateral continuity, stratigraphic position, marker beds, and if these characters are found to match, in case of rock units which are widely separated from each other, they are correlated on the basis of lithostratigraphy. So, the studies in case of lithostratigraphy involves the following. Litho units are identified on the basis of beds, members, formations, and their mappability is established with respect to 1:50,000 scale. The rock compositions such as color, texture, composition, structure, mineralogy are studied over multiple locations to establish lithological similarity. Sedimentary features such as current bedding, graded bedding, ripple marks, shoal markings are studied to establish the right side of character or reverse position of the strata. Thickness of strata and their lateral continuity, contacts with upper and lower formations are duly studied. Presence of marker beds, rock units with distinct lith character in the rock succession are identified. For example, volcanic ash layer, coal seams, conglomerate bed. These are some of the unique rock successions, and if they are there, they serve as the marker horizon and they help in lithostratigraphic correlation. Because these are rare, they establish the identity of a rock formation. That's why these marker horizons are of importance in lithostratigraphic correlation. Similarly, the presence of distinct mineral assemblages such as zircon, epidote, garnet, magnetite, which are the heavy minerals, they identify the rock units with uniform lithology and thus throw light on the provenance from which they have been derived. Provenance is the source rock from where these sediments are derived and deposited in the sedimentary basins. If these heavies are being considered, the heavy minerals are being considered, if they are found to be same, that means these sediments have been derived from a common provenance, and thus these heavies, they help in the process of correlation. Besides these, the unconformity surfaces, if present, they are of much help in establishing the correlation. If the unconformities are not there, in that case the vertical rock columns are compared, borer logs are found out to know subsurface position of rock units, and then the correlation is established. Besides this, the study of change of phases that take place laterally is also studied. For example, a sandstone bed grading into shale seaward is interpreted as a phase change within the same time plane. So, sandstone and shale, they will be considered to be same unit, same litho unit, because one is grading into the other, and there is it can be said that there is no break in sedimentation. The change of phases are common in marine transgression and marine regression. This is common in transgression and regression. So, lateral continuity will be considered to be the same time plane in which they have been produced. They should not be considered as separate units. In case of marine transgression, there appears to be the deepening of the marine basin. So, pure limestones which occur in the deeper part of the oceans are found to occur with marly sediments. What is marly sediments? They are mixed carbonate clastic sediments. Similarly, in case of marine regression, there appears the shallowing of sea basin and the marls occur with clays and shells with sandstone, marine sediments with continental deposits. So these features will have to be considered if it is a case of marine transgression and marine regression phases. So after all these studies, the data relating to lithology, vertical thickness, order of superposition, stratigraphic position, they are combined to build geological cross sections for the multiple locations, and then they are correlated if the lithology characters are found to be exactly identical. For example, the Deccan Troughs. The Deccan Trough basalts of western India and central India are correlated with respect to distinct basalt flows and intertrappean beds. Because we know Deccan volcanism has affected the entire southern India. So we find the volcanic flows in central India and in western India. If we correlate them and also correlate the intertrappeans, then the lithological identity can be fully established. Because we know the Deccan Troughs, they evolved over 65 million years to 60 million years back over the western India, western and southern India. And the volcanism took place through a number of phases. And each phase is found to spread over a time period of 30,000 years. That's why the volcanism that has taken place in western India and central India, they can correlated if their lithological characters match exactly, then they can be correlated on the basis of lithostratigraphy. Similarly, in case of Gondwana coal formations of Jharkhand, West Bengal, Son, Damodar, Narmada Valley, Mahanadi Valley, Godavari Valley, they are correlated with respect to their lithology. For example, sandstone, shale and coal sequences. Similarly, the Cretaceous chalk formations of the entire world, because that is related to marine transgression. So, the Cretaceous chalk formations or the pure chalk limestones deposited in marine environments are correlated all over the globe. And what are the limitations of lithostratigraphic correlation? Limitations are the rock types can change laterally through phase change. As it was discussed earlier, that sandstone may grade to shale during marine regression or during marine transgression. So there we have to consider that sandstone and shell to be time parallel, or the same, they are deposited in the same time plane, and they should not be considered as two distinct litho units. So that limitation is there. We have to take into account whether it is a formation that has been produced in the marine transgression condition or in marine regression condition. In that case, it is seen that lateral continuity is there between the sedimentary formations. Sandstone may grade to shell, shell may grade to limestone. So that is one kind of limitation, because as I have already told that lithostratigraphy is based on lithological characters only. If we consider sandstone and shell which are produced in the same time plane to be two separate units, then the correlation, the very purpose of correlation will fail. So that is a type of limitation. Similarly, the same lithology may develop at different times. That means they may not be always time equivalent. As lithostratigraphic correlation is based on purely on lithological characters and exactness in lithological characters. So the stratigraphic correlation will fail there if exactly same lithology is found to develop in different areas in different ages. And when you talk about correlation, correlation must be equivalent in age. Correlation must refer to equivalence in age. They must not differ widely. Lithology, in case of lithostratigraphic correlation, we take into account the lithology, no doubt, but that does not mean that they will vary in their age widely. In that case, it will not be a process of correlation. So that's why similarity in lithology may develop at different times, and that is not always time equivalent, but they will be correlated on the basis of lithostratigraphy. So that is a type of limitation. Then let us discuss about biostratigraphic correlation. So in case of biostratigraphic correlation, we take into account the fossils, the age of fossils into account. Fossils of the biozones. Biozones are noted for their fossil content. So to achieve this, we have to remember the law of irreversibility of biological evolution. What is this? It states that the organisms cannot evolve into the preceding forms even if they find themselves exactly similar conditions in which their predecessors lived. Thus, each assemblage of past life preserved in reflects a specific stage of evolution of life. These stages are confined to specific periods of geological time, and therefore fossils indicate the age of the biozones which are considered for achieving correlation. So in case of biostratigraphic correlation, we determine the age of the fossils, and on the basis of that age, the litho units are correlated, or the biozones are correlated. So while considering the fossil assemblages in biostratigraphic correlation, following facts are considered. The organisms may represent the biocoinos and their environment may be biotope, which represents the living organisms in a biotope. there are second group of organisms which are fossils, which are fossil organisms. So in case of biotope and biocoinous, there are living organisms, collection of living organisms. But when you talk about thanatocoinous, in that case, we take into account the fossil organisms. The fossils that may be derived from the neighboring distant biotope and that may be mixed with the inhabiting species and their fossils, or the death assemblages of the fossils which are obtained from the nearby biotope and those which are found in the inhabiting environment, inhabiting biotope. They together, their fossils constitute thanatocoinous. So, the in situ assemblages in thanatocoinous, they are really helpful in correlation. So, thanatocoinous should be studied with respect to the in situ or inhabiting species. There are third group of fossils which are derived from older rock formations by erosion and sedimentation process to a biotope. These are called as allochthonous fossils or derived fossils. These fossils are not useful in correlation because they do not indicate the age of the rock formations in which they are found. They give a incorrect age of the rock formation in which they are found. That's why these allochthonous fossils, they are not useful in biostatic correlation, and thanatocoinous fossils, which are the representative of that biotope, they are useful in correlation. So, you have to establish by the biostatic correlation, the biozones are studied, whether these are taxon-rich biozone, concurrent biozone, lineage biozone, opal biozone, assemblage biozone, interval biozone or acme biozone. These are studied, and to determine these biozones, the first appearance datum FAD and the last appearance datum of each fossil in a rock formation are determined, so that the rock unit with respect to the appearance, with respect to FAD, with respect to LAD, they are determined, so that the litho unit can be exactly dated. The equivalent biozones from different areas are matched to find out correlation. And mostly the index fossils, they are of much help in establishing the biostratigraphic correlation because they are noted for their short geological range and wide geographical distribution. Similarly, microfossils, they are also very useful in correlation because they are mostly found in marine environment. And the requirements for good biostatic correlation are there must be accurate identification of fossil, use of rapidly evolving and widely distributed organisms as is the case with index fossils, and the continuous sedimentation must be there with no major gaps so that the fossils can be evaluated properly, correctly, and use of microfossils wherever possible for finer resolution in biostratigraphic correlation. Example, the ammonites, they help in correlation of Jurassic group of rocks in India and Europe. Similarly, Globigerina species help in correlation of tertiary rock formations all over the world. Now let us discuss what is chronostatic correlation. It is a process of correlation based upon matching of the rock units on the basis of their absolute age. More or less absolute age. Not relative but more or less absolute age. Here lithological characters are not taken into account. So absolute age of the rock units are found out by taking into account the radiometric age dating, by taking into account the index fossils, by taking into account the eustatic change of sea levels, by taking into account the volcanic materials, tephrochronology, by taking into account the paleomagnetism imprinted in the newly formed rocks as are found on both sides of the mid-oceanic ridges. So, correct dating of rocks are based on these studies: radiometric age dating, fossil study through index fossils, eustatic changes in sea level, tephrochronology and paleomagnetism. So in case of radiometric age dating, the radioactive elements such as uranium, rubidium, potassium, strontium, they are studied and their half-lives are taken into account, because it is known that the radioactive elements that decay series are not found to be affected by any change in temperature pressure conditions to which the rock bodies are subjected to. And that's why these radioactive elements contained in igneous, sedimentary and metamorphic rocks, they are being studied, particularly in igneous and sedimentary rocks. They are not found to be affected and their decay constantly goes on in a definite rate, and therefore these radioactive elements, they indicate the exact age of the rock formations and thus help in correlation of the rock units. Similarly, the fossils, they are also of help in detecting the age of the rock types, particularly the index fossils. For example, if you take into account the index fossils, for example, trilobites, they represent marine Paleozoic formations. Similarly, Glossopteris ganotis vertebraria, which are the plant fossils, they indicate the Permo-Carboniferous age of the continental formations. So, there are certain fossils which are highly diagnostic, highly indicative of the age, exact age, they are useful in correlation. Similarly, the dating of rocks on the basis of radioactive elements are very, very useful in tracing the exact age of the rock types, and thus if the age is being matched, then correlation is possible. Besides this, we have the eustatic changes in sea level, and these changes in sea level affect the rock types globally. That's why they are of help in correlation. Eustatic changes are the rise and fall of sea levels, and these are caused by variation in ice volume with ages during glacial period and interglacial period. During glacial period, there will be marine regression. In interglacial period, there will be marine transgression. Similarly, thermal expansion and contraction of seawater, or change in volume of ocean basin due to sea flow spreading, because the mid-oceanic ridges are the sites where new oceanic rocks are being created and the ocean floor expands sideways. In that case, there will be, that may lead to eustatic change in sea level. Marine transgressions and regressions occur simultaneously worldwide. These events create boundaries, maximum flooding surface, that is in case of marine transgression, and the sequences thus produced in their boundaries, which are isochronous, and hence serve as chronostratigraphic markers useful in correlation. For example, worldwide marine transgression that took place during late Cretaceous time, which produced the chalk formations of limestone deposits of Europe, North America and India, which are all found to be correlated on the basis of these limestone formations. There is another process by which the age can be determined, that is tephrochronology. Tephra, that means layers of volcanic ash or pyroclastic material that are ejected from volcanoes and are deposited over large areas at a time surrounding the place of volcanic eruption. As recently it is found in case of Ethiopia, the entire European and African countries and parts of Asian countries, they are found to be deposited with the tephra produced from that Ethiopian volcanic eruption. So these volcanic eruptions, they produce tephra and they are found to be distinct with respect to their geochemical characters, and they are found to be deposited in widely separated areas over hundreds and hundreds of kilometers away, and they are found to be preserved, and they are indicative of a particular age. They are isochronous. So over thousands and thousands of kilometers, these volcanic materials as pyroclastic materials, they are found to be distributed by airborne transport, and thus they establish the isochronous nature of those materials. Each volcanic eruption produces tephra with unique geochemical fingerprint. This point will have to be remembered. Unique geochemical fingerprint. Every eruption is noted for its own geochemical characters, which is distinct from others. So that is chemistry, trace elements, isotope, minerals, they are all distinct, which can be traced across wide regions, and the radioactive elements contained in them serve as absolute time markers and hence useful in correlation. For example, Toba ash materials erupted from volcano of Indonesia, 74,000 years old, are found in South Asian countries, Indian Ocean and East African lakes. Similarly, Campanian ignimbrite of Italy, 39,000 years old, it is found to spread over Europe, Russia and North America, that help in their correlation. Then paleomagnetism. Paleomagnetism refers to the study of magnetic polarity displayed by the magnetic minerals, particularly on both sides of the mid-oceanic ridges. The volcanic materials are always found to erupt along the mid-oceanic ridges, and the sea floor spreads away from the mid-oceanic ridges on both sides, and the eruption is found to be distributed equally on both sides of the mid-oceanic ridges. And these rock types, they contain in them the magnetic minerals which record in them the Earth's magnetic field. Earth's magnetic field is found to change its polarity with time. With a finite period of time, the Earth's magnetic polarity is found to change. It may become normal or it may become reverse. In the present time is the present magnetic normal polarity is being shown by Earth, which has been there over the last 0.7 million years. That is Brunel's normal. So survey of ocean basins around the mid-oceanic ridges have located stripes of volcanic basalts that present magnetic normals and magnetic reversals. through their magnetic mineral alignment. Hence, through these magnetic minerals, the oceanic crust of the world are found to be correlated. Similarly, the rock types of different ages, Precambrian rocks, Mesozoic to Paleozoic or Cenozoic rocks, are found to be correlated through fossil magnetism. Because the magnetic minerals that develop in a rock contains or it aligns itself along the Earth's magnetic field, and if you take into account the distribution of the rock types or the distribution of the continents throughout the geological time, it is known that the continents have united and have been dismembered several times in the geological history of Earth. And the last unification of continents was in form of Pangaea, and the dismemberment is going on. Dismemberment has already been over, and all the continents are going to face another phase of unification in coming 200 million years, because one major supercontinent cycle lasts for about 400 to 500 million years. Already 250 million years have passed over the last breaking, and therefore it is expected that all continents will come together by another 200 million years to form a single landmass. So, the magnetic studies, the magnetic minerals which are produced during different periods of time, they contain in them the magnetic minerals aligned along the Earth's magnetic field. And on the basis of their orientation, magnetic inclination, magnetic declination, it is possible to determine the age of the rock types. And through that, it is possible to correlate the rock types. For example, Brunner's normal, it shows 0.78 million years to present. Matema reversed, it shows 0.78 to 2.58 million years. Gauss normal polarity 2.58 to 3.58 million years back. Gilbert reversed 3.58 to 5.33 million years back. So, if, for example, if a sediment layer shows the Brunner's to Matema transition, this transition, Brunner's normal to Matema reverse transition, that means the rock type must be belonging to this age, 0.78 million years. So it automatically correlates with 0.78 million years everywhere. So by establishing the normal polarity and the reverse polarity, along with study of their age, it is possible to correlate the rock types all over the globe with respect to the magnetic scale. Now let us have a comparison of these three: lithostratigraphic correlation, biostatic correlation, chronostatic correlation. What is the basis? In case of lithostratigraphy, basis is the lithological characters and not age, and there must be similarity in rock types, not in age. In case of biostratigraphy, fossils are taken into account and biozones. So they are time equivalent. In case of chronostatic correlation, the absolute dating of the rock types is made. What is the objective? In case of lithostratigraphy, the objective is to identify and match the rock units with similar lithology which are found in distant areas. In case of biostratigraphy, to correlate the strata based upon their fossil content or biological time interval is established. And in case of chronostratigraphy, the objective is to find out the equivalence of the lithology on the basis of their absolute age or on the basis of the fossil contents. What are the tools? In case of lithostratigraphy, tools are study of rock types from mapping, from boreal studies, from geophysical studies, geophysical logging. In case of biostratigraphy, the index fossils are studied, faunal succession of biozones are studied. In case of chronostratigraphy, the radiometric age dating, magnetic reversal study, tephrochronological studies are carried on. What are the units that are used for correlation? In case of lithostratigraphy, formation, member, bed, they are being used. In case of biostratigraphy, the biozones are being used. In case of chronostratigraphy, the system, series, stages are being used as the units. The nature of correlation, surface. In case of lithostratigraphy, it may be time parallel. It may not be time parallel, but lithology must have to be same. Lithology must have to be repeated at different times about the earth. But in case of biostratigraphy, that approximately time parallel based on fossil evolution. Chronostratigraphic, it is exactly time parallel based on isochronous surfaces. Dependence on environment. Lithostratigraphy is strongly dependent upon the environment of deposition. Biostratigraphy is less dependent on environment because the organisms can migrate from one place to another. And in case of chronostratigraphy, it is independent of environment of deposition. It is purely based on study of the time of the formation on the basis of the radioactive elements, magnetic elements, fossils, or by study of tephra. Limitations. In case of limitations of the chronostratigraphy, in lithostratigraphy, the limitations are the same lithology may be found at different times in different places. In case of biostratigraphy, the fossil may be absent in igneous metamorphic rocks or in non-marine environments. So, in that case, correlation will be difficult on the basis of biostratigraphy. In case of chronostratigraphic correlation, it requires accurate dating of rock types and the study of global markers as indicated by the tephrochronology. So, these are the major differences between these three types of correlation, and I have tried my best to explain the correlation in the best possible way, and I thank you all for your patient hearing to this talk. Thank you all.